Hello, everyone. My name is Alexis Mitchum. I'm a PhD student here at CMS working in Dr. Maya Breitbart's lab. And today I'm going to be presenting my research on analyzing the spawning patterns of economically important fishes on the West Florida shelf through DNA barcoding of fish eggs. So a pretty common question that we get a lot is, why study fish spawning, right? Why is it important? Well, we know that reproductive timelines and spawning locations of fish species on the West Florida shelf are largely unknown and are pretty data deficient. But we do know that these habitats support a huge variety of marine life, including commercial and recreational fishes. So fish eggs are a good proxy for gauging spawning patterns, but they are a little bit challenging to identify just because we can't identify the species just looking at them with the naked eye alone. So we have to use some molecular tools. So the sample method that we use to actually collect the fish eggs is called the CUFES, or Continuous Underway Fish Egg Sampler. And I've included an illustration here of what the CUFES actually looks like. So it essentially takes in water from the hull of the ship, brings it in through the system that you see on your right-hand side. And that's called, it's what we call the daiquiri machine. So it essentially shakes all the eggs around and essentially just discards all the excess water, runs it through a flow tube and onto the filter, and we can just collect the fish eggs straight from there. So for those of you who may not be familiar with DNA barcoding, has anyone in here ever heard of the technology 23andMe? Everyone, right? So if you've ever heard of 23andMe, you're already a little bit familiar with the technology that we're using for this project. So I've made this illustration to break down DNA barcoding in 4 easy steps. So step 1, we collect and sort the fish eggs from the West Florida shelf. We extract the DNA of each individual fish egg. We amplify the DNA using a method known as PCR. And then we use the raw DNA sequences to find a species match within our database. So here's a map that I've included of all of the locations that we have selected for DNA barcoding of these fish eggs. So this cruise took place from August 28, 2022 to September 26 of 2022. And as you can see, we have a really good spread here on the West Florida shelf. So all of this area here consists of the West Florida shelf, all the way from Pensacola down to the southern tip of Florida. So we do have some results from these 2022 fish eggs. So 623 eggs were collected in total. 507 of those eggs were successfully barcoded, which is about an 81% success rate. 406 of those eggs could be identified down to the species level, with 36 unique species identified. So 191 of these eggs came from 7 commercially important species. And I've created this illustration to scale of the abundance of each species that we found. So of course we found the most scattered species with 148. We also have some fan favorite species like 4 species of snapper here, as well as blackfin tuna as well. So how are we going to improve the 81% success rate? It's pretty good, but it could be better. So we do have a little saying that likes to circulate through our lab, and that is, the biggest room in the world is the room for improvement. So the question that we're asking is, are we missing species just due to primer bias? So a primer is essentially a small piece of DNA that helps amplify a section of the target DNA. They attach to conserved regions of the DNA, and they help amplify regions with different sequences for each species. So we were testing 2 target primers for this particular study. So we reran the mitochondrial cytochrome C oxidase 1 primers that we usually use for these studies. We're also testing a new one using 16S rRNA primers. These primers have been shown to amplify things from bacteria to rats and even fish. So we'll be testing this new primer as well. The CO1 primer is good because it is a relatively conserved gene between species. So a little breakdown of how we actually ran this experiment. So we took 75 eggs that originally failed through our first PCR. We tested their DNA concentration using qubit. And with these, we had 22 eggs amplified with the CO1 gene, and we had 53 that amplified with the 16S rRNA. And I will note that the 22 that amplified with the CO1 also amplified with the 16S rRNA. Now, in total, we had 28 eggs from this amount that was actually successful. Now, we had 11 from 16S rRNA that were successful and 17 from CO1 that were successful. And that's about a 37% recovery rate for these bad eggs. So as you can see, we got ourselves into a little bit of a rabbit hole with this portion of the project. So I made a list of pros and cons to actually doing this fine tuning. So the pros, we had one species added that we did not identify with the first round, and that is Prionatus carolinus, the northern sea robin. 28 sequences were added to our database, and this increased barcoding success rate from 81 to 85%. So pretty good, right? The cons to this fine tuning were that it was a lot of work with very little reward. We only got an extra 4%, which you could, I guess, weigh as a pro or a con. But it does double the cost that we're originally paying just by running this one time. And as you can see, we had more species that were identified using CO1 primers than 16S rRNA. We had 17 with the CO1 and only 11 with the 16S rRNA. And this is because the 16S rRNA database is just not well developed for fish species yet. We found that the CO1 primer database is more well developed for this purpose. And finally, doing the 16S rRNA gene comparison is just less comparable with previous studies as we've used CO1 primers in the past. So what's next for my PhD project? So we did have some technical difficulties in the year of 2023. The CUFES broke on us midway through the track, twice actually. So we're pivoting the project to ask new questions about the Gulf of Mexico. More specifically, we're going to be looking at the yellow-edged grouper. So the yellow-edged grouper has become a prevalent importance for many organizations such as FWC, and it's a more elusive and rare species and is actually thought to be a deep sea species. So we will be looking at the section known as Pulley Ridge off the southern tip of Florida. We found some yellow-edged eggs there, and we will be looking at stations around that site to see if we can identify some more. And the time series will continue in 2024 with a novel seasonal component, which I will be adding as a part of my research. So we'll be expanding sample collections from annually to quarterly throughout the year to identify some new species that may not just be spawning in summer. Thank you.